Welcome back, students. Dr. D.A. here again. And in this brief presentation, we're going to talk about how information is transferred and used in the cell by bringing it from the information that's stored in DNA into other molecules that are called RNA, and then into the sequences of polypeptides, which ultimately are the way that we say genes are expressed. So please have your PowerPoint notes ready so we can go ahead. We're going to start by talking about the molecule of RNA. So far, we talked about DNA, or deoxyribonucleic acid, and how the information for functioning of cells is stored in there and can be transferred when the cell duplicates by doing what we call DNA replication and copying the sequences of DNA that were present in the original cell. Now let's talk about this other molecule called RNA. So RNA, or ribonucleic acid, differs from DNA in several things. Okay, and here's some of the key differences between them. First of all, it contains a different monosaccharide residue. Remember, DNA uses the so-called deoxyribose. RNA uses ribose. RNA contains the bases A, G, C, and U, or uracil, instead of A, G, C, and T. So whatever function thymine or T played in DNA, that function is exerted by U in RNA or uracil, and it pairs with adenine. The other difference is that typically RNA exists as a single-stranded instead of a double-stranded polynucleotide, and that is in general. Okay, it's not always, but in general it exists mostly as a single-stranded molecule. Now, uh, if I give you, for example, a podcast or an audio file, you can probably listen to it and write it down in words or request what we call a transcript. It's essentially a converting of information from one medium into another one, but still in the same language. In other words, in this case, that would be English. So one of the things that happens is that the information that is contained in DNA is going to be transcribed into RNA. In other words, it's the same information, it's the same language, but just a different medium that we're going to use. Uh, Again, we're going to have different enzymes involved. This one is called RNA polymerase. It's going to attach itself to a strand of DNA, and there is a particular sequence of bases in DNA that we call a promoter site. When the RNA polymerase attaches there, it disrupts the double helix so that it opens it up, and the sequence of deoxyribonucleotides on one strand is going to be used as a template for making RNA. Now, notice that unlike replication, where both strands of the DNA were copied, in this case, only one strand of the DNA is going to be transcribed. And that strand, we're going to call it the template strand. Once more, as we see here, the DNA double helix that we see at the top is going to be opened up by specific enzymes. Notice that the RNA polymerase is going to begin at one end and copy the information that's on the template strand of DNA. Again, it's going to be in the 5' to 3' direction. And as it copies it, the nascent or new RNA strand starts kind of peeling off. Notice again that we're only copying one strand of the DNA, and we're not making an exact copy in the sense that the new strand of nucleic acid we're making is not DNA anymore, it's RNA. You can see in the bottom picture here that the mechanism is very similar to the one we saw before. The growing RNA strand is going to start at its 5' end and copy bases according to the complementary code that we talked. So T gets copied into A, C gets copied into G, A gets copied into U. Remember that RNA does not use thymine. It uses uracil or U. And again, at the 3' end of this ribose unit here, you're going to have an ability to connect to the phosphorus here that's attached to the 5' carbon of the incoming nucleotide triphosphate, and then a double phosphate set here. The pyrophosphate is going to be taken out. Another interesting feature here is that there are actually 3 different types of RNA, and they have different functions. So messenger RNA, abbreviated as mRNA, they may be of variable sizes, and it depends on the gene and the protein to be manufactured. Messenger RNA is going to carry the information that specify what protein is going to be made in the next stage of the process. This message is carried as a sequence of nucleotides that is complementary to what was copied from the template strand of DNA, remember? All right. There is another kind of RNA called transfer RNA or tRNA. These are typically small molecules of RNA, usually 73 to 93 nucleotide residues. And what they do is they are going to bring in an attached amino acid that can be added to the growing polypeptide chain as the code is translated from the mRNA into the polypeptide sequence of the next stage. The other type of RNA is called ribosomal RNA or rRNA. These are relatively big molecules of RNA, and there's different types of them, and they combine with other proteins to form a structure inside the cell that's called a ribosome. This is a multi-subunit complex in which protein synthesis takes place. In other words, it's kind of like the factory where proteins are made using the information that the messenger RNA is bringing from the original genes in the DNA. In order for this to work, there has to be some kind of a code, and we call it the genetic code. And during the 1940s and 50s and 60s, this was one of the major thrusts of molecular biology research. It was trying to define what was the genetic code. How does the information in the DNA of the cell get converted into specific sequences of amino acids in proteins? So the information that mRNA carries regarding the primary structure of a protein is read 3 bases at a time. In other words, the genetic code consists of sets of 3 bases at a time. Each series of 3 bases is called a codon, and it specifies a particular amino acid. So because it's 3 bases, we call this a triplet code. Now, if you think about some math, if you have, you know, this possibility of having, 4 possible bases in the first position of the codon, 4 possible bases in the second one, and 4 in the third place, that's actually 64 possible combinations. There's 64 ways of combining bases in this manner. But remember we said that only 20 amino acids are used to make proteins. So that means that in the genetic code, some amino acids are going to be coded by 2 or more codons. In other words, the genetic code has some repetitions in it. Here is a typical format of the genetic code. And as you can see, what we've done is we've said, okay, on the left side of the column, the left column, I'm sorry, we have what could be the base at the 5' end, U, C, A, or G. And then the second position, which is the one in the middle, you can see on the top, U, C, A, or G. And then on the far right column, we have the third position. This would be the 3' end of the codon. This is another version of the genetic code in which is built in kind of like a circular manner. And so you can see in the center circle, you have the U, C, A, G going clockwise. And then for each one of those, you have 4 possible bases on the second tier, and then 4 more on the last tier. So in our class, we're going to use this more standard table format. Let's see how it works. Let's say that your first position on the codon is an A. Let's say that the second position on the codon is a G. And let's say the third position is a C. As you can see, A, G, C, if you read it from left to right, which is 5' to 3', it's going to correspond to the amino acid serine. Notice that right above it, there's another possibility, and that would be AGU. In other words, that codon also codes for the amino acid serine. As we said, some amino acids have more than one codon that corresponds to them. There's also some specific sequences that we have to take into account. For example, methionine, which is produced by the codon AUG, is the initial or start codon for every polypeptide that is made in the cell. In other words, every messenger RNA, when it goes out with the sequence, the protein building factory is going to be looking for this AUG sequence, and that tells it that I'm sorry, while I was recording this presentation, I had a glitch here. I think my computer kind of died out. I hope the video is okay, so I'm resuming it here. Hopefully there's no problem with it. I'm sorry about that. As I was saying, the protein synthesis machinery of the cell will scan the messenger RNA until it finds this sequence AUG, and that tells it that's where the polypeptides start, and the first amino acid that's going to be inserted is going to be a methionine. We also have some other special codes. For example, the codon UAA, the codon UAG, and the codon UGA. All three of those codons are stop signals. So when the protein synthesis machinery is scanning along the RNA and making proteins, when it hits one of these codes, it's a signal for it to stop. And that's where the polypeptide will end at that point. And we'll talk more about that in a few moments. Now, in a higher organism, such as, let's say, human cells, where the DNA is contained inside the nucleus, the messenger RNA has to be transported from the nucleus into the cytoplasm of the cell. Now, also in the cytoplasm is going to be the transfer or tRNA. So what the tRNA does, it has a series of 3 bases that is complementary to a codon on the mRNA. And we call this sequence an anticodon. So what's going to happen is the enzymes that are making proteins will bring in the tRNA and match the anticodon and the codon by base pairing to ensure that whatever amino acid is being brought by the tRNA is added at the proper place in the growing protein chain at the correct time. So there are particular enzymes called tRNA synthetase enzymes that are able to match the corresponding amino acid to the corresponding tRNA. So these are other enzymes that make sure that when the tRNA and amino acid complex is made, it matches the correct amino acid to the correct anticodon. Here are 2 different descriptions or representations of tRNA. On the right side is what it might look like as a molecule. And you're showing here different sections. You can see that it's kind of like a bit of an L-shaped kind of thing. And basically what you're going to see is at the bottom part, in this picture is kind of in red. Over there at the very bottom, that's where the anticodon is going to be. On the left side is a more popular way of describing the tRNA. It doesn't represent the way it looks like in three-dimensional space, but it's something called the clover leaf representation. And you can see that there are portions of the RNA. You can see here, for example, at the top, this blue sequence here, this little yellow sequence here, this little green sequence here, and this pink sequence. All of those are sections where the RNA has some base pairing. In other words, some double helical structure, kind of similar to the one in DNA. There's also a few loops in here, and at the bottom of this clover leaf structure, we can see what is called the anticodon. The anticodon. Notice that the way it works, this comes from the 5' end over here. So the anticodon, remember, has to be read in the opposite direction. So this would be the anticodon for an amino acid that is specifically phenylalanine. At the top, at the tip of this blue section, which would be the tip here on the far right of the three-dimensional structure on the right, that is where the amino acid, in this case phenylalanine, would be attached by the tRNA synthase enzyme. And so this tRNA will bring that amino acid onto the growing chain, and it'll be added wherever the corresponding codon is on the mRNA. This is another representation of it. Again, you can see over here on the bottom right where the anticodon is. This is a sequence of 3 bases that will match the corresponding codon in the messenger. There are several loops here in the structure, and at the very end here is where the amino acid is going to be added so that it can be then transferred to the growing protein chain. Now, excuse me, in order for the information to be converted into protein, it's almost like you're changing to a different language. So in transcription, you change medium from DNA to RNA, but it was still the language of nucleotides. So we call it transcription. Now we're going to convert the information from the language of nucleic acids into the language of polypeptides. So this is a new language, and because of that, we call this process translation. In the next in the next presentation, I want to talk about this, and I'm going to talk a little bit about what are some of the ways in which genetic expression can be modified or regulated. Thank you so much.